Hello, my name is Dave Douches. I'm a professor at Michigan State University in the Department of Plant, Soil, and Microbial Sciences. And today I'm going to talk to you about the biotechnology work that we do in our potato breeding and genetics program. And so at Michigan State University, I lead the potato breeding and genetics program. I have a great team of staff and students that help us create genetically improved potatoes. And so we tackle it in a couple ways. One is through conventional breeding, where we make crosses and try to select for potatoes that have resistance to diseases, insects, viruses, and abiotic stresses, and also trying to target the market quality traits. And for us here in Michigan, that is actually chip processing. And but we also, through our conventional breeding program, take our biotech crops to the field to look at their economic value. And so we've been in our lab developing biotech potatoes. And so my philosophy here at Michigan State is that I really feel it's the integration of conventional breeding and biotechnology that kind of creates the best opportunities and have the most positive impacts for the potato industry. And what I'm going to talk about today is first some of the biotech work that we're doing that has an influence on Michigan and maybe the U.S. market. And then I'm going to talk about our USAID or USAID project, where we are developing late blight resistant potatoes for some target countries outside the U.S. So first of all, the potato breeding program started getting involved in genetic modification around 1994. And that was about the time the private sector also got involved. And we saw the release of a genetically modified BT potato that conferred insect resistance. That was around 1995. There were other events that came forward through the Monsanto company, and those were removed from the market. We won't go into that story. Then along came the J.R. Simplot company, and they developed a biotech division, which they call Simplot Plant Sciences, and they worked on developing potatoes that were genetically modified that only had potato genes in them. And they were called innate potatoes. And the first ones were released in 2015, and their different events are on the market and being grown commercially. There is a PVY resistant spunta in Argentina, but there really isn't a lot of biotech potatoes out on the market compared to some of our other field crops. And so Michigan State University has been working on this for a long period of time. And we've been able to, in the past year, get receive non-regulated status for some of our genetically modified potatoes through the USDA APHIS. And so I'm going to just give you a little background on that. One of our potatoes is been improved for cold storage so that we can store the potatoes colder and they will not accumulate reducing sugars. And we achieve that by silencing the vascular acid invertase gene. This is not new technology. It's been around for a while. It was work that was demonstrated in potato by Jiming Jiang when he was at the University of Wisconsin. He's now at Michigan State University. And so by silencing the vascular invertase, you prevent the conversion of sucrose to the reducing sugars, glucose and fructose. And glucose and fructose, when fried, will give a browning of the potato chip. And so you can see that in the picture here. Here's our variety Calcasca, and here's and you can see how they're brown. And then here's the potato chips that were from our invertase silencing event, and you can see that they chipped quite well. And this was after 6 months of very cold storage. 40 degrees Fahrenheit is 10 degrees colder than we would typically store the potato. And the reason we did this work was that we had a variety called Calcasca that had high yield, and a good tuber type, and had scab resistance. It grew well for the farmers, but when they put it into commercial storage, the sugars went off. So our strategy was, we were saying, with this invertase silencing, maybe we can fix the potato and make it a marketable potato for the potato industry. And so to do this, we went to the USDA APHIS, and they've recently updated their regulatory system. Excuse me. And so now the process to really achieve non-regulated status has been simplified because of their taking into account their 30 years of experience of working with biotech crops. And so this process is called the Regulatory Status Review, or RSR. And so on these next couple slides, I have the information that is required to get an RSR submission and review. And so I'm not going to go into the details of this. Sorry about that. Oops, wrong way. But the most important thing is you have background information, but we have to then supply molecular characterization data of the potato event that we're looking for a non-regulated status on, and then provide a description of the trait. And if there's any kind of unique information, we also send that along and send it in for review. And so we did that, send it in 2023, and then in 2024, the last day of January, we received non-regulated status on our potato. And so we're quite excited about that. As a land-grant university in the public sector, we feel this is a great milestone, because this is really the first vegetable that's reached non-regulated status in the U.S. from a public research institution like Michigan State University. So we're quite excited. So what does this really mean? Well, when we have this potato, it can be stored colder and longer. And that's important to the farmers, to be able to provide potatoes to the processors over a longer period of time that are of quality for processing. In addition, we see that it reduces a stem end defect, because stem end defect is due to a release of reducing sugars in storage. And as I said, it extends the life of the potato. And so what it really does is it provides stability for the supply of potatoes. And that is really valuable in a year-round business. So that quite exciting. So in Michigan, potatoes can be harvested and processed essentially from October all the way till the 4th of July. And so essentially 11 months of potatoes can be supplied out of Michigan. Our second trait that we've been able to achieve non-regulated status is what we call Xerico. And this gene was brought to our attention because it gives us drought tolerance. But we also found out that it increases the starch content in the potato. And this is a gene that was cloned by a researcher here at MSU. It's a MSU patented gene. And so we transformed this into potato. We saw that in greenhouse conditions that we did have some drought tolerance. So we then decided to take those plants to the field. And we've run trials between 2019 and 2023. And in these replicated field trials, we've seen very good data. First of all, if you look at this figure I have here, so here's Ranger russet, which is the non-transgenic potato. And then here's 3 events from Ranger russet. And so what you see is in this top here is the yield. So the big question is with abiotic stress genes is if you express them in the crop, will you have a yield penalty? And we did not see this yield penalty. So that was very exciting to see. There may even be a little bit of a yield advantage, but measuring yield is can be very challenging. So I'd rather not stick my neck out, but saying that at least we're not having a yield penalty, but maybe there will be a yield advantage. Secondly, was the increase in starch content. So for us, this was a real surprise. We have our hypotheses why the starch content has gone up, but it was a very significant increase. So you see here 1.080, which is kind of what the industry considers a threshold for processing, and we were able to raise them essentially like 7 or 8 points on the specific gravity scale. So that's a very significant amount of starch increase, and that's a benefit to the industry because less oil is used in the processing of those potatoes. So we're quite excited about taking this to potatoes that can be used in the chip processing market and get them on the commercialization track. So we sent in this potato for RSR and we were able to get approval in October of 2024. So we're quite excited that we have a second product to work with. So having this, we see that this has the potential for reducing irrigation needs and then higher recovery in the chip processing plant. And also this trait really has no effect on the processing quality traits. It seems like it's a neutral trait from that perspective. So we're going to continue to create new events and take these to the field for the potato industry. And so the U.S. regulatory system also has the FDA and the EPA. Well, the Food and Drug Administration, FDA, has a voluntary review. We're planning to do those reviews for our potatoes as we collect the data. And the EPA, or Environmental Protection Agency, because we're not working with a pest, these traits are not under the purview of the EPA. So our track to commercialization is clear at this time. So what I'd like to do now is go into our USAID USAID project. And this project, I'm actually the director for that. We call it the Global Biotech Potato Partnership. MSU is the lead institution. The International Potato Center is our major partner in this, and we work with other universities. I'll talk about that in a minute. But our goal here is to deploy stacked R genes for late blight resistance. Okay, I'll explain more about that. So this partnership right now is a 5-year multi-institution cooperative agreement. Between USAID and MSU, but the International Potato Center is our very strong partner in this. Their operation that we work with is in Nairobi, Kenya. We work with other universities like the University of Idaho, and we're working with the national research organizations in our target countries. Our target countries are Bangladesh, Indonesia, Kenya, and Nigeria. There are other countries that we would like to target, but at this point, these are our 4 countries to work with at the moment. What we're trying to develop is late blight resistant potatoes, and we're also looking to develop other resistances in these potatoes for the future or for the pipeline. So, and our goal is to get these potatoes to the smallholder farmers. So it's not just developing them, but is taking it to the field and getting it in the farmers' hands. We have a great team that's put together. That we have our core team here at Michigan State, but what's important is we have to bring together a lot of expertise for this. So Mark Galan is a biotechnologist at the International Potato Center and kind of is a deputy director with me. But it's important that we have a global regulatory affairs lead. We have a global seed deployment and stewardship lead, global pathology lead, Then we have global and molecular technical leads and a communication lead. So there's, and of course we have a project manager. So there's really key people that need to work together to move this partnership forward. So I haven't really spent any time talking about potato. I've been working on potato breeding and genetics my whole career, but why potato? Well, potato for some people you may not realize, but it's the third most important global food crop that's consumed by humans. Okay, there is actually more maize production, but that's used in many purposes besides feeding humans. So we fall in behind wheat and rice. There's been a great increase in potato production over the past couple decades, and to show the importance of the potato in feeding people. And most of this increase has come in developing countries. From an economic point of view, potatoes are on a different scale in terms of international trade. It doesn't move around the globe as much, so we don't have as much volatility in the pricing of potatoes as other world food crops. And for some people, you might be surprised, but today potato is very nutritious. It's a good source of fiber. It's high in potassium, vitamin C, and B6. And so the UN quote is quoted here as the potato produces more nutritious food more quickly on less land in harsher climates than any other major crop. So here's a global map, and you can see the countries that produce potatoes. Potatoes are grown just about every country in the world. You can see the top countries here, and you look at those top six, and nobody would be surprised in those. Well, maybe some people might be surprised by China, but potatoes feed people, so they grow. India and China grow a lot of potatoes. But after Germany, look at that, Bangladesh, number 7 as in world production. But the country of Bangladesh has a large population on a small land mass, and so they need to feed a lot of people, and they grow potatoes in the winter as an rotation with rice during the summer. So a very, very important crop. Look at that. It's almost as much as Germany. It's almost half of the US. And then our other 3 countries that we work with are Kenya, Nigeria, and Indonesia. So why late blight? Well, late blight caused by Phytophthora infestans is really a worldwide problem and causes billions of dollars of damage. And in the US, we control these our disease, and in Europe with good management with fungicides. But in developing countries, they may not have the resources or whatever, and they have an environment that's conducive for late blight, and so it's a threat to food security in some of these developing countries. And where it's very tropical and there's a lot of moisture, the amount of fungicide sprays needed is extensive and can be quite costly, and it and also has an environmental and human health exposure issues associated with it. So if we can create late blight resistant potatoes for potato production, that has many benefits to society. And this map just shows where late blight can be found. So where potatoes are grown, you usually have late blight. Some places worse than others. So, okay. And despite breeding efforts for many, many decades, we're still relying on fungicides to control the disease. It's been difficult for potato breeders to develop late blight resistant potatoes. I'll show you a slide on that in a minute. And as I've mentioned, there's limited resources to purchase fungicides and protective equipment in developing countries, and this leads to fungicide issues that impact the health and environment. So what's really exciting for me is that over the past 25 years or so, there's been a lot of work to understand the resistance to late blight. Uh, and and what we've learned is that there's a series of R genes that can be found in potato, R genes or resistance genes. And what these genes do is they don't actually attack the pathogen or produce a chemical that attacks the pathogen. What they do is these R genes recognize an effector in the pathogen and then turn on the plant's natural host resistance. So we refer to this as effector triggered immunity. Very, very fascinating. So if an R gene can detect that the late blight is landed on the leaves or the stem, the plant will defend itself. But the pathogen Phytophthora infestans is a really good foe and can evolve and avoid detection. And but there's only so many ways that can avoid detection. So there's a lot of R genes that have been cloned, and they have different effectors that they can detect, or they have the ability to detect different effectors. So if we can stack a series of R genes, we can together we can combat the majority of the Phytophthora around the world. So taking a step back about the breeding, so this is a demonstration of conventional breeding to develop late blight resistant potatoes. So in 1959, not very recent, there was they knew that this species Solanum bulbocastanum and the Solanum acali had resistance to late blight. So through crossing, you can follow this chart if you want and see the crossing procedure that over time they were able to combine the resistance genes from those two species and put it into a background cultivated background. And in 2005, they released two varieties, Bionica and Toluca. And so that only took 46 years. So not that to me is not really something to be a way to continue to breed potatoes. It demonstrates how long and hard it is to get resistance. And they only got two R genes in, okay? So we're in the biotech era. So let's take advantage of the tools that we have. So actually, if you go back a slide, I say here over 70 R genes have been identified and mapped, and more than 50 have been cloned, okay? So there's a whole database of these R genes that are available now. And a lot of this work in cloning R genes was done in the Netherlands at Wageningen and also in the UK to make these genes available for research. So, okay, so with biotechnology, we can kind of avoid the downfalls of traditional breeding where we have to back cross in from a wild species a resistance gene. We know that in traditional plant breeding, when we back cross in from a wild species, we're going to bring along a lot of undesirable genes. So if our yellow is our desired gene, green is our undesirable, we've got those tagging along. So it takes rounds of back crossing to get rid of them. But with biotechnology, we have our desirable gene. We can insert that right into the potato and not have any undesirable genes along with it. Makes a lot of sense to me. So work's been done to say, okay, if we deploy a single R gene, it seems like that will be overcome by the pathogen quite quickly. It'll have the opportunity to evolve effectors that avoid the detection by the plant, by that R gene. So here's one way of looking at it. So we have this plant that has an R gene in it, and inoculate it with 5 different strains of late blight, and it holds up to most of them, but that one, it went down. Okay, if we look at another R gene, we see a different pattern of susceptibility and resistance. And so you can see here it killed, weakened here, strong here. But then if we put 3 R genes together, under all conditions, we're seeing very healthy plants with no infection. So we've taken this strategy. Let's stack 3 good R genes and deploy them in farmer preferred varieties. And so we did a proof of concept of this at MSU in 2017. These potatoes here in this field picture are nice and healthy green, and on the borders you see dead potato vines on both sides. Well, the border rows are susceptible variety. There's been no fungicide applied to this field, but it was inoculated, about a month or more prior to that, and so you can see that our plants stayed very healthy green in the field, and foliage has continued to grow and produce good yield underground, whereas our border rows are dead. So we're taking this concept to our target countries, and USAID is supporting this, and we call ourselves the Global Biotech Potato Partnership. We have 5 objectives, So one is to demonstrate this commercial potential of our 3 R gene events in the field in our target countries. Second is to collect the data so that we can submit a regulatory package to those countries for the general release of those 3 R gene late blight resistant potatoes. And then while this is going on, we also need to develop a seed deployment system and a stewardship of those biotech potatoes. And to feed the pipeline, we feel it's important to not just rest on that original 3 R gene potato, but to develop a next generation of disease-resistant potatoes, as well as developing in-country capacity in the process of developing those second-generation potatoes. And lastly, having communication and advocacy. So we have 2 different 3 R gene constructs in this first generation. We have these 3 R genes, and if you see right here, one of them is Solanum bulbocastanum, and down here you can see the 2 of them are from Solanum bulbocastanum. So that's that same species that they worked with in 1959, but these genes have been cloned out, and now they're stacked into a single construct, and we have them in different varieties. Here we have them in Diamant and Granola, and here we have them in Shangi and Victoria. Okay, so at Michigan State University, we worked with Simplot Plant Sciences to create that first generation of 3 R gene potatoes. They're a great research institution that's trying to develop commercial potatoes, and so we worked with them to develop those first potatoes. When they created their events, they have the resources to actually test the functionality of each of the R genes that were in those events that were generated. And so from those 130 events that were tested, they selected the top 10, and those top 10 came to Michigan State University. So they had the pathology expertise and also the genetic engineering expertise to work with to get to that stage. But something interesting, just from a transgenic trait point of view, when you only have a single R gene and you're trying to find one that has a good level of resistance, most of the events will not have a high level of leaf blight resistance. You might find a few that are quite good, but if you have a stack of 3R genes, the majority of the events that you pull off will have a high level of leaf blight resistance. So there's clearly an advantage out in the field, but in actually just generating events, you generate more events to work with, which is important in selecting good events that have the agronomic characteristics you want. So we've got some slides here of the different trials in our target countries. Here's Kenya. There's Mark Gilan and Eric Magambe from the International Potato Center. And what I like to say is that we don't need, well, we run field trials. They're under replicated conditions with good experimental design, and so that we can run statistics. But the results we get in these trials, you don't need statistics to see that the resistance is working. Here you can see the leaf blight resistant potato, and here you can see the potato that is not resistant. Quite a difference. In Bangladesh, one of the preferred varieties by the farmers is Diamant. So we transformed that. And in 2023-24, we had confined field trials in Bangladesh. And you can see results here of these might actually be the granola potatoes. And then in Indonesia, we have the granola variety, which is dominant in their market. And we've been running multi-location trials there, confined field trials, for a number of years since 2021. In Nigeria, they actually also grow Diamant, and they also do grow some SIP varieties, Victoria, Shangi, and Tigoni. And so these are being tested, and you can see here in the picture that quite black and white. The green are the survivors in the field, those plots, and the ones where they're brown is where the potatoes have succumbed to leaf blight. In Kenya, it's the same story. We are not testing Diamant or granola in Kenya, just the SIP varieties, which are very preferred in their markets. Along with the field trials, here's our global lead pathologist, Phil Wharton, from the University of Idaho. He's quite involved with confined field trials at the different sites, and as well as helping with pathology work and developing the data collection procedures. I'll come back to Phil in further down the presentation here. And so here's just an overhead view of the trials in I would believe in Kenya. And so when we run these trials, we also like to bring out the farmers and the dignitaries to demonstrate to them. It's when you can see it in person, that the difference between a resistant and susceptible potato, it's like a miracle that you can plant these potatoes and survive, and they survive under the intense leaf blight pressure. In Kenya, we've already done a socioeconomic analysis, and this data has been recently published. And what we've learned through that socioeconomic analysis, that there's great economic benefits. That so when you kind of look at all the data, basically the bottom line is that the annual benefits, if they were to grow the 3R genes Shangi, is over $8 million a year. And if you prorate that out over 30 years, it's almost $250 million of economic benefits. So there's a real advantage to this. I've seen some of the preliminary data on the socioeconomic analysis in Indonesia, and it's actually double of what the Kenya benefits are. Here's Nigeria. The trials there have been going well. It's again black and white. Here we're looking at the figure, and what this is showing the late blight severity. And so here's where we're seeing the all the severity is in the non-transgenic potatoes. So the potatoes are staying quite clean in the field, whereas our check varieties, non-transgenics, are actually completely killed. In Indonesia, that's one of my favorite places for our field testing, because the tropical conditions there, late blight is present from the time the potatoes emerge in the fields till they're harvested. It's in many other countries, late blight kind of moves in during the season and attacks the crop. Here it starts right when the potatoes are emerging. It's just that tropical conditions are just so conducive to the pathogen. And so we feel that we can get the most intensive test in that country. And our granola event, UG 234, has been performing very well there. And here you can see the results of the granola. Again, we're not looking at statistics here. We're just looking at the picture. If that wasn't statistically significant, I don't know what would be. What else would be? So here you can see what happens in the field after just 68 days. These plants are barely surviving, whereas our 3-argine event is quite happily growing along. Bangladesh, I was there last February to see the field trials just prior to harvest. We had the secretary from the Minister of Agriculture come out, and she was quite happy to see the results of these confined field trials, and is supporting the effort to move these potatoes through the regulatory system. So we're quite excited to see that. And so this is the Diamant variety. Again, we don't need statistics here. Okay, but to put together that regulatory dossier, we need to put together a lot of data. And so what we need is the agronomic data. We need multi-location trials from 1 or 2 seasons. We need to collect that data. We need it exposed to late blight, and so protected from late blight. We need to collect tissue to do compositional analysis. So we collect tubers, send them off for compositional analysis, collect that data to make sure there's no changes in the food quality. We need to do the molecular characterization in the lab. That was some of the work we did at MSU to get our potatoes approved. And we got to just make sure that the integrity of the insert is good, that it ideally is a single copy insert, that there's no rearrangements, there's no insertional effects, and there's no vector backbone that got inserted into the genome randomly. The regulatory agencies want some protein characterization. This is very difficult with argines. Argines are really not producing a large amount of protein. There's really trace amounts. So it's not like a BT gene that's producing a lot of protein that you can then extract from a plant. So what it's come down to is that the Simplot Plant Sciences was able to demonstrate, based upon weight of evidence, that argines have a history of safe use, because argines are natural in plants. Their mode of action is not producing any specific chemical. It's more of a detection, and they're also very specific to specific pathogens, and so and that there's really minimal expression of these genes in the plants. So based upon that these argines should be safe to consume. Okay. So coming back around to Phil Wharton, here he is in Indonesia collecting late blight samples. And so we're working with a disease. This disease can evolve. There's a lot of genetic variation. And so what Phil is doing is in each of our target countries, he's collecting isolates from around the country and understanding what is the diversity of the pathogen in that country. And also over time, as we do that, we can see if there's any changes in the pattern of diversity as we deploy our late blight resistant potatoes in these countries. So it's very, very important information. And so it would be really good to know if there was a breakdown of resistance, what isolate was causing that breakdown in the field. Uh, our our fourth objective is that second generation of disease-resistant potatoes. And so the technology keeps advancing, and one of the persons that we can honor for that is Jonathan Jones at the Sainsbury Laboratory in the UK. He's really devoted a lot of his career to studying and cloning many R genes in potato, and he's made these R genes available for us in the project, through his institution. And so our second generation potato has two new R genes that we believe are very strong. We're packaging it with the VNT1 gene, which was in our first generation, because this gene seems to be a very strong gene. And then we're combining that with virus resistance, one to potato virus Y and the other to potato leaf roll virus, which are the two most important potato viruses in the world. If we want to maintain seed quality, we need to control late blight, and we also need to control viruses. And so this will really improve the resistance package of the potatoes for the second generation. To me, that increases the sustainability of these potato varieties and for their use in production. And so as a proof of concept at Michigan State University, we generated some events with new constructs. So this one had the 3 R genes plus the PVY resistance in detached leaf bioassays. The cardinal variety here was completely destroyed, whereas 3 events here were standing tall. And we also tested for virus resistance. This is ELISA stick test, and so you can see if the potato plant is infected with virus, it'll form a band here as it gets wicked up this strip test. Okay, and so here is a non-inoculated cardinal, and then here are the 3 events that were late blight resistant, and you can see they were also PVY resistant based upon that strip test. Yeah, but I forgot to say we did inoculate those potatoes with the strain prior to to that test. And so what we've been actually able to do in the US is we've been able to take our first generation 3 R gene potatoes and also some of our second generation potatoes and receive non-regulated status for those potatoes. So this to me demonstrates for those developing countries that our country regulatory system approves them, and that we also, it makes it easy for us to test these and produce seed in our country for our target countries as the project moves forward. The last thing to bring up about our next generation is targeting a really challenging trait. Bacterial wilt resistance has been needed in potatoes. Despite the use of wild species, they really have not been able to find any strong resistance to bacterial wilt. And so this has been a challenge for the potato farmers in tropical regions. And through the biotech approach and Jonathan Jones, they've been able to clone some R genes for bacterial wilt resistance. So a combination of 3 R genes for bacterial wilt resistance were transformed into potato and tested at the International Potato Center, and we're seeing positive preliminary results. So really we have a 3rd generation of potato that can combine late blight resistance, have the virus resistance, as well as the bacterial wilt resistance. That would be a sustainability package that is would be phenomenal for the farmers. So what's also important to us in this project is that capacity development. That's part of Objective 4. And so our global leads are working with our partners in our 4 countries, and we're working in these areas of the product development and technical support, disease resistance management, the regulatory affairs, socioeconomic and market analysis, and the seed sector and stewardship, as well as communication and advocacy. We want our scientists, partner scientists in the target countries to be able to be self-sufficient when our USAID project ends. And so lastly, we have communication. And so that if you can't communicate the value of the product, and also be able to talk to people at the different in the different areas about the product, it's going to be hard for people to take on these potatoes. We know that genetically modified crops are a concern to some groups of people, and so we need to make sure that people understand what we're doing, what the product is, what the value of it is. Are there any risks? What are the benefits? So that that people are comfortable with this technology. What we see is that the farmers are quite excited to adopt this technology. We also, but we need to make sure that the governments and the consumers and people in society accept this technology to create better potatoes. So the way we kind of summarize our communication strategy is we proactively communicate technology benefits while passively monitoring anti-movement activity, and promote a transparent and fact-based message with personalized audience-specific framing, while partnering with our biotech allies for consistency and amplification of efforts. It takes our whole team for us to have good communication. So trying to sum things up on our USAID project, well, we've been running the field trials in the country with those first generations. We get complete control of the pathogen. We're evaluating the diversity of the pathogen in these countries, and we actually interestingly see a lot of difference in the diversity across our different countries. That's a seminar in itself. The molecular characterization of the lead events have been completed, and that's contributed to the dossier development for the regulatory approval. And so this slide says that we're going to complete the research and have the approvals in the next couple years. So let me just go to the next slide here. So I want to say is that we actually in Kenya are in the process of submitting our dossier to the Kenyan government for review. So that's quite exciting. And what we expect is that Indonesia will in 2025 and possibly Nigeria will follow behind, and then Bangladesh would probably be 2026 when we would be submitting a regulatory package if all goes according to plan. So coming back to our accomplishments, the 2nd generation disease resistant potatoes, that work's being done. I feel that that's really important to have that pipeline. You don't want to develop a technology and then just sit on it the advances that keep coming along, it would be really hard to ignore the value of the technology as it moves forward. And what's important is that we strengthen the capacity in our target countries. The scientists, are coming to Kenya at the International Potato Center for training, and so that they can go back to their countries and work on that. And so just this morning, we had a meeting with our Bangladesh colleagues, and they're actually have events on these second generation potatoes coming out of their experiments now. It's quite exciting to see that. And I don't think if we didn't have the capacity development, this wouldn't be happening. And then lastly, this project really cuts across, a lot of important themes like the environment, the gender issues. We really didn't talk about that, but there's we're working with the women farmers and with the women scientists as in our target countries, and we've talked about the socioeconomic importance to this project. And with our communication, really, we have a network that ties together 20 different global institutions in 5 countries to have that communication message. So what in the next 2 years, if we get these potatoes in the field, we're saying kind of moderately 90% fungicide reduction. There are other diseases that attack potatoes, so you might need fungicides, but late blight is the major one, so we're saying 90% reduction, so that reduces input costs, it reduces crop losses, it reduces exposure to fungicides for the farmers, and we get those higher yields, so that can also mean food security for some people. But it also, if they get higher yields and reduce costs, that's potentially more profitability on the crops that they grow, which should improve their livelihood. I think this is one way that we can help uplift the farming communities in our target countries. There's many other things that can be done, but this is what our skill set gives us, and we're able to work on our countries. So just to close this off, you can see the message here. And so I'd like to say genetic engineering is one technology that responsibly increases the productivity and economic standing of smallholder farmers while reducing inputs and environmental impacts. Or you can simply say that our late blight resistant potato is a safe food product that benefits the environment and the community. So we're quite excited to be working on this project, and we're going to be really excited when the potatoes are approved and handed off to the farmers in our 4 countries of Bangladesh, Kenya, Nigeria, and Indonesia. So I appreciate you listening to my talk here, and I don't have the ability to take questions, but if Cholani, who's running the class, is able to take questions, she could always pass them along to me at some future date. Thank you.